Good afternoon, everybody, my dear friends. I am Mrs. Neeta Maitre and will be presenting a PhD pre-submission seminar on use of VSTs in gene prediction of cotton genome, a clustering perspective, under the guidance of Dr. Manali Shivsagar, professor at Ekatra Chauhan College of Engineering, and currently she is principal at Sri Datta Medi Polytechnic College, Nagpur. I am from computer. Yeah, I am from Computer Science and Engineering Department. Let's go to the agenda. Agenda is we will be seeing introduction, research objectives, literature review, genetic prediction or finding, blast search, cotton, what exactly the cotton is. We will be going through the slides later when we will be going over this agenda. Let's see the introduction. The introduction starts with data mining, a very well-known word in the field of computers. Just try to apply it on the biological background. So generating knowledge from a given data, the stepwise general strategy that we could see from the diagrammatic representation is understanding the domain, identifying the source data, selecting data, pre-processing it, then applying the actual data mining, post-processing, and then coming to a real task problem which we want to solve. So why data mining? Because the field, as you could see from the name of my topic, the data which we will be dealing with is biological data. So data mining is preferred over data analysis just because the biological data is tremendous, large in size, complex, and it needs new and sophisticated applications in order to perform different tasks. So basic biological data mining tasks which we can summarize are searching to look for a similar pattern. Second is classification or clustering, to create groups in a given data set. One is based on supervised, another one is based on unsupervised. Query search, given a query, searching a pattern into the literature for a given data set. So introduction of bioinformatics, as we all know that it's a multidisciplinary field which is consisting of molecular biology, genetics, computer science, mathematics and statistics. Sequence analysis plays an important role when we talk about biological data and biological sequences. Knowledge of data mining can be very well applied by performing sequence analysis. So, when we talk about bioinformatics, the central dogma everybody might be knowing, having the words popped up and seen: DNA, RNA, and protein. So these are nothing but the basic central dogma of molecular biology, which is based on three different processes. One is transcription, another is translation, and the third one is replication. So DNA replication, synthesis of RNA from DNA is transcription, synthesis of proteins from RNA is translation. Sources, so let's have a look at biological data. Biological data is available through many sources. The ones which we have referred is National Center Biotechnological Information, that is NCBI. And the three basic data sources that we have referred in our research work are GenBank, DBEST and Unigen. So GenBank is a primary sequence database which is consisting of DNA sequences. DBEST consists of it is a division of GenBank, rather the superset is GenBank, division of GenBank, which relates with the express sequence types or cDNA, and the Unigen, it is nothing but the clusters of the VSTs which are present. So based upon this background, let's have the research objectives. Research objectives is first to identify various factors involved in gene finding or gene prediction. Gene finding and gene prediction are simultaneously used in documentation when it comes to for gene prediction, to validate express sequence tags as the resource in gene prediction. So when we say that gene prediction, we will be doing it with ESTs. Let's validate whether it is correct or not with the help of already existing things that we will see later on. To use short length EST approach instead of DNA strand in gene prediction. As Dr. Shrisagar ma'am's research doctoral work was based on 9-step gene prediction process. Now, we need to propose, and we would like to propose a short and sweet approach towards the gene prediction. So that's why we have gone for the ESTs as the resources. Next comes the systematic approach to perform clustering of biological data set. A novel approach to reduce number of computations to find similarity of sequences. There are various sources there are various already existing tools which perform these similarity of sequences, but now we want to propose a new one which will be doing it for us. To study various data sets of cotton, that is Gaussian. Later on we will be explaining why we have chosen Gaussian as the data set which we will be under consideration. So we have to find a simple way of handling large biological databases as called as EST. Next is a clustering algorithm which will help in coding region prediction. The coding region prediction is basic requirement for when we talk about a gene set of doing a gene prediction. So coding region prediction, what exactly the coding region prediction is and everything we will be looking at it later on. Ultimately what we want to provide is nothing but the optimal solution for sequence similarity once a query sequence is given to us. So literature review, many papers were reviewed, few of them, rather only few of them I have mentioned it here, just because these I found the most relevant to us. The diagrams drawn here are not the ones which are given in the literature as it is just the interpretation of what I felt about it instead of writing it in words. So making sense of ESTs by clubbing. The clubbing is cluster on the basis of similarity. It is going to have, it takes a list of sequences, then it has having based upon high scoring pairs, 3 different types in which it is considering. It's a very basic kind of algorithm which tells the condition, merges into the cluster and creates a new cluster on non-satisfaction of the condition. The observations on it is based on high scoring pairs. It is robust, efficient, it is written in Perl, and special programmers are needed if you want to do anything into it. Next is novel identical clustering algorithm approach, which is based on basic K-means algorithm, which is the heart of whenever we talk about clustering, we talk about K-means. Here it is, they have given a different approach or different way of looking at K-means by giving a new value to it. Say for here the choice of centroid is made crucial. It is random selection of centroid which we used to do earlier in case of normal K-means algorithm is avoided. They have given imposed some set of conditions in order to choose the centroid for the initial type. Third one is CD8. It is nowadays used. It is a fast program for clustering the things. It is based on shortcut filtering and it generates cluster. We will be having slides which will compare our approach with CD8. Next is gene prediction or gene finding. As we know, there are 2 types of organisms. One is prokaryote, another is eukaryote. The gene structure is simpler and smaller in size. Genome is smaller in size as compared to the eukaryote in prokaryote. Now, when it comes to cotton is from the eukaryote based cotton. So here in this case, there are no introns when we talk about prokaryotes. But here, when we talk about eukaryotes, human beings are also eukaryotes. Exons are interrupted by introns. Why exons? Why introns? All these things will get clear in next few slides. So prokaryotic gene finding and eukaryotic gene finding types are there. Then we are having rule based, content based, similarity based, and ab initio based. Similarly, we have eukaryotic gene finding where rule based, content based, feature based, similarity based, and combinational approach. So now feature based and combinational approach, these are the 2 additives when we talk about the eukaryotic gene finding. We will see where exactly it differs. So gene prediction method. So gene prediction is grossly considered as the coding region prediction, where a region in biological sequence is identified which will code for protein. So ab initio gene prediction is you are going to check out for a particular set of pattern. Say for example, a start codon and a set of end codons. If we are getting that pattern, we will try to recognize it as a coding region. Coding region is the region which will map ultimately to a protein at the end. So ORF finder does it for us. So start codon is a triplet of codons which initiates the process of translation, and end codon is the set of codons which stops the set of process of translation. So ATG is recognized as the start codon, and there are three end codons. One is TAG, another is TA, and third one is TGA, which can be considered as stop codon. Then next is homology test gene prediction, wherein we are going to check out whether the evidence or existing data set is there for can I have an homology of the query sequence with the already existing data set. If we see that EST word comes into picture here, so whatever we have done or we have proposed, it is a homology based clustering perspective to the gene prediction. Consensus based gene prediction is it is going to have perform various number of programs together, and the combined approach will fetch us to the predicted exons and retain it. So now express sequence tags, it's a one shot sequence for the whole library of complementary cDNA or complementary DNA. So it is called as Poor-Man's genome. Now let's have what exactly the human genome is and how EST is. So genome is consisting of a big sequence, the size might be 670 MBs. It is consisting of introns as well as exons. Now when it comes to ESTs, exons are only present. The ESTs might be considered as an erroneous ones, but still it is going to be instrumental when it comes to gene prediction, because ultimately coding region is the one which is nothing but exonic in region. So exonic region is always can be considered as tending to gene prediction. Non-exonic regions or intronic regions are not the coding regions. This is the way in which EST can be formulated. Tissue, you are getting an mRNA, cDNA, and at the end of the cDNA we are getting ESTs. The EST library is created based on this. So uses of ESTs, full length cDNA can be generated. As we could see, cDNA is going to give you ESTs, so ESTs can be generated. From ESTs you can generate full length cDNA. Genome analysis, it is rapid way of expressed genes. Expressed genes are the ones which are going to at the end lead to proteins when you are talking about a set of codons there. Gene expression studies and identification of new genes. New genes which is going to have certain different functions which are not yet uncovered. Now that is considered under the new gene sequence. For doing all these things, we need a representation of the file format, and that file format in bioinformatics is generally a FASTA format. It is a text-based representation. It is very easy to understand because it deals with only 4 words: A, T, G, and C. Allows representation of both nucleotide and peptide sequences. As we could see, we have seen rather DNA and protein. So protein and DNA, these are the two types of ways in which we can represent. So ESTs are represented in FASTA format. This is the general example of a FASTA format where you could see the gossypium is nothing but the cotton bead, cDNA it is nothing but the EST, and now this is the whole sequence which we can have. The speciality of EST sequences is its length is very limited, that is from 200 base pairs to 700, limited to approximately 200 to 700 base pairs. On the contrast, the genome size is mostly 800 MBs and all, as far as cotton is concerned. So BLAST search is the traditional way of doing the things. BLAST stands for Basic Local Alignment Search Tool. It is again developed and maintained and managed by NCBI. It's a very good tool which finds similarity between the nucleotides and the protein sequences, and it is mostly for genomic and protein comparisons. There are various options available for BLAST. Those are the types of BLAST are BLAST P, BLAST, and MEGABLAST. These are the comparisons between them. When you talk about amino acid as a query and protein database to be searched on, we will be going for BLAST-P. Here in this, in our work, we have used BLAST-L. Let's see why we have chosen cotton. As everybody knows, cotton has a highly valued crop. It has a global importance, high farm gate value. Once it is coming out of farm, it is surely going to have high farm gate value. The conditions that are suitable for the growth of cotton are hot and dry weather. We also know that because in Nagpur we also go for cotton and all. Cotton producing countries are China, India, United States, Pakistan and etc. These are actually the top 4 that I had mentioned. Biology of cotton, its biological name is Gossypium. It is having 13 chromosomes. The Gossypium is composed of more than 50 species distributed widely all over the world. Major traits include these 5 things. The data sets from all these 5 we have taken for our research. The scope for research is, if you could see, Unigene is a database which is consisting of the clusters. And now, if we see the current state, we don't have any cluster present for Arboreum, Barbadans, Arboreum. You don't, and if you talk about the EST sequences available, 63,642, still you are going to get only 8 clusters, and under which you are covering only 26 sequences. This prompts us to get something into clustering in order to generate clusters for any number of sequences under any circumstances. Our choice is clustering just because to get insights into the data. For that, we need to pre-process the data. General applications of clustering. Clustering is just getting the similarity and keeping the dissimilar objects away from the clusters. So our algorithm, we named it as mid-clust-pi. As you could see, the name itself says the mid portion of ESTs for clustering and implemented in Python. Most of the bioinformatics tools and techniques which we use here are developed in Perl. Perl needs specific programmers, and that's why we thought to have an option which would be having which could have the easy to understand kind of language and usage. So this is specifically the algorithm is implemented in nature. We will be seeing how it is. It is a homology based clustering, as I had mentioned before also. It is also called as an evidence based because ESTs are the evidences which will prompt you to get the genetic position or the coding region position. So it needs pre-processing as it's a part of data mining. Let's see what exactly the pre-processing is. Pre-processing is input EST sequences, arrange them in descending order, extract the middle portion, create an upper triangular matrix, and make. So input sequences. This is our main algorithm where we have generated the mean and median. Threshold is set to mean or median. We have created an additional vector called as row ID and max value which we consider for the particular column. Sequence 1 is considered as the first cluster. Then a clean cluster representative file is generated. The next sequence is taken. This is just a simple incremental algorithm, but it has not having the draw. It is providing you the facility not to skip any of the sequences and give the perfect cluster identification approach. So distance matrix is generated to utilize the computation. The choices are given, mean, median or user defined. The value will be compared to threshold, and threshold can be system defined or can be user defined. Now this is as I had already said, CD-HIT EST is a comparable program, and it is nowadays in boom. It's a web server which gives you a package of n number of comparisons which we could get. So the previous approach, the original approach BLAST, is nowadays replaced by CD-HIT EST, and which is greedy in nature and is not that way accurate as compared to our approach. So the way in which we could compare is CD-HIT. It's not implemented yet. This is a comparison chart wherein the only one thing is missing, that is our approach is going to go for a dynamic programming approach, and CD-HIT is going to use a greedy approach. So dataset uses 500 species from every 500 sequences from every breed, and then a combined sequence database was formed. The implementation background consists of Python 3.6, features of some Python we have used and the pandas version. The validation of ESTs, whether ESTs are the correct ones to go for the for computational purpose, yes. That's why we have gone for using ORF against the ESTs and then blasting them. The results are like this. The algorithm that is followed is accept an EST sequence, blast against its gene and test okay or not. It is a similar kind of thing. Similarly, for clustering, we have used an incremental algorithm, which we have the overall clustering algorithm consisting of pre-processing and the whole algorithm. How the query check is made? The query can be put in, the clustering of matclust5 will be done, the comparison of the query sequence with the cluster representative will take place. Once you get a match, the candidate clusters will be checked. If you are not getting a match, then in that case no match found. Otherwise, you are going to proceed with the candidate and again checking for the threshold. The results. Validation of use of express sequence times as a source for computational purpose. 96% of the records under consideration has shown the results compared to the potential ORS. Out of remaining 4%, 39% cases which were not having ORS. So even if we see 97% results favored that ESTs could be our instrumental source as compared when it comes to the potential gene production. This is the graph that we get when we talk about validation of ESTs as a resource. Here 263 records we have gone for the specific breed. Next is we could make the complete EST can be checked out against the middle part of ESTs. Just because if you talk about complete sequence, it can also be helpful and the middle part can also be. But if you want to go for an optimal, the middle part fetches you the correct solution as well as a very less time. The graph itself tells you that these are not visible I guess, but the ranges that we could see is time ranges are 300, 600, 900, 1200 and these are in seconds. So we have done it for the we have shown it for the representative ones. The actual ones are very large. Cluster formation from ESTs. Now middle part of ESTs is considered to be accurate. The 95% of the number of clusters which are found out for mean are same for the medians, and more precisely the time required for clustering in most of the cases mean is more than the median. So this is the way in which we got that median versus mean threshold. So results summary, 86% cases of the results are matching with BLAST. BLAST is the well established kind of or tool which is provided by NCBI and which is more frequently used by the researchers. 92% cases results are matching with their own query the way we have implemented mid+5. If we see the accuracy of our algorithm is more, 6% cases the query cover was matching, they are not matching with the top 5 elements of the hit table, and thus we could see the results of clustering. The mid+5 are compared to the complete sequences also, but there is only 5% of the deviation. Now the advantages of our approach is sequence processing is limited to very small number of base pairs, but still the results generated are almost in accordance with whatever the well-established BLAST gives you. For very large dataset, sophisticated machine configuration, it will surely save time because clustering is only a one-time task. Then, unlike BLAST, standalone BLAST, we have also implemented standalone BLAST, which is going to give you every time a new time for searching. So this kind of variable kind of output you won't get. Whatever you will be giving query for our approach, you will be getting the same kind of output every time. Programming for bioinformatics application is mostly done in Perl, and if we see the Perl code, it is quite a bit complex. On the contrary, the Python gives you a very user-friendly kind of output. Disadvantage could be query check module need to use output of mid+5 for generating the final result. It could be merged into the one single integrated system, but we felt that it should not be done like that because there might be some set of applications where we are going to use only need clusters of the dataset. And clustering, though proved to be an efficient way of handling large data set, but clustering time is an additive if you talk about a small data set and operating on it. Applications of this kind of work, the prediction of diseases is important. We have written one paper which has gone for checking out the infection in cotton. So this infection, which is given by Pantoia agglomerans, it could not be seen when you are getting the ball of the cotton growing. But if you go for this kind of, making out the similarities between Pantoia agglomerans, you will be in a position to get the DNA sequences mapped, and then the infection can be detected. Similarly, gene therapy and in agriculture to improve crop yield. Recently, the BT cotton was a great achievement as far as this thing is concerned.
Please conclude.
Yes, last slide. GMO and non-GMO. The clustering technique is applied to biological data set of cotton. The metrics of performing the clustering is analyzed. The implementation taken into account the total number of clusters generated. Whatever, the work could be easily applicable to the field of agriculture in identifying plant diseases, increasing crop yield, and so also GMO, non-GMO kind of issues can very well be handled if we talk about this kind of thing. These are the publications, few references. These are the book chapters which I had referred, and the books for implementation in Python, BioPython, bioinformatics. Thank you.
I think I must say that if you were my student, I would tell you straight up. The presentation is quite chaotic, it's not systematic. As a student of computer science, you are talking about algorithms, you are not talking about time complexities. Forget computer science, even as a basic research presentation, it is very disappointing that you don't know what it means by citation of the reference. Do you know what it means by citation of the reference? In the beginning, you were mentioning that you have mentioned that is your work is based upon your supervisor's work. Okay, but you could not cite that. You could not say that. Okay, the name of the authors, the year, and this. You know there is a difference between presenting something for a class or teaching somebody a tutorial or as compared to a research defense or a presentation. Do you know the difference between these two kind of presentations?
Yeah, whatever I had done, I need to express this considering that.
It's not that whatever I have done. No, there is a lot of thought has to be given to a systematic arrangement of the. So you have to start with what was the literature, what is the present state of the art, and in that what is the motivation, what is the observation of your literature, what is the motivation by which you are observing that this is called as a research gap analysis, and based upon that, then you may say that you are bringing out a proposition. Once you are bringing out a proposition, that proposition it has to be you just can't say that it's better anyway. You need to have a significant number of other approaches and methods, and you have to systematically compare and evaluate those methods, and then you may have implementations. In the implementations, what is the data? The data has to be systematically arranged and said that this is the characteristic of the data, and then when this data is applied and the various different methods in computer science, we look at computation complexity. You are talking about a presentation in computer science without having even mentioned the word computation complexity. Okay? If it is a clustering algorithm, it's a data mining approach. So there also, algorithm. Can you please tell me how do we actually present or define an algorithm?
Algorithm is the systematic way in which you perform a
What
certain
are
task.
very important aspects which are actually defining an algorithm?
I mean the input process and the way.
See, input, output, apart from that, definiteness, finiteness, correctness. As a person of computer science, if you don't mention the definiteness, finiteness, and correctness, then it's not an algorithm. It is not at all acceptable in a kind of a formal presentation or in research work and things like that. I've seen this paper also. This whatever it has been through the like there also is I can't really read and understand that what is really the contribution, what is really the algorithm, what is the processing approach. I was thinking once you mentioned that okay class and all that. So see if it is a pattern recognition approach, I will look for what is it, how do you find out the features. Okay. Once we are finding out the features, so then based upon the features, how many features have been found? For the given data, how many features are found? Okay, what is the algorithm by which we are able to get clusters? What is the identity of the clusters? What is the composition of the clusters? How many clusters? How do you choose how many clusters? Okay, so there are so many questions which are there which you are simply breezing through everything, but there isn't really any answer for those kind of questions. I'm not just trying to put you down or anything. I'm saying that based upon what I have seen, no. See, nobody really gives till now. I've seen nobody really gives presented the references as bullet points. Nobody gives. Either they give name here, okay, or else they give a index number. Okay, in my opinion, if somebody has not learned this referencing approach, then I don't think so they are actually eligible for being awarded.
That will make those changes. Actually, yeah.
See, more than making those changes, you realize that there is a problem with the attitude.
Yes.
In research, we are trained to be methodical in writing, in presentation. We don't want wrong conclusions to be drawn. We want the right conclusions to be highlighted. We take pains to explicitly say that this is the methodology. There should be repeatability. With this methodology, we can obtain these results. The writing has to be systematic, so when it is communicated, somebody else can also understand and reproduce those results.
Yes, sir.
Okay, fine.